in India. So next to this, we discussed about the history, the scope, opportunities, then constraints in the cultivations. So how these uh, medicinal plants can be cultivated. So we are discussing about the ashwagandha. Today we discuss the ashwagandha plant detail, its cultivation, what are the chemical composition, important medicinal use, or how this can be used for the curing the various diseases. So ashwagandha, it's a local name. Withania is English name, or we can say the um, vernacular name of that plant. So the importance and chemical composition. So now screen is visible. So importance and chemical composition. This is the ashwagandha plant. So ashwagandha having the botanical name Withania somnifera. For your objective type question, all the botanical name, its family, then chemical constituent, whatever constituent is available in that plant is essential. So uh, you can make the MCQ at yourself. So all the botanical name of all medicinal plants and aromatic can be write on one page. Then its family, then important constituent, or we can say a medicinal property, or which important um ingredients is present in that. It can be asked for the MCQ type question or your semester examination. So you can write it down on a same separate page. Okay, so it's commonly known as winter cherry and Indian ginseng in English. Several types of alkaloids are found in these plants, out of which the vithanine and somniferine are the important chemical constituent is present in the ashwagandha or the withania plant. In addition, the leaves are important to contain the 5 unidentified alkaloids. So other than this, many alkaloids are present in this plant. The total alkaloid content in ashwagandha roots Indian type has been reported to vary between 0.13 to 0.31. So these are the percentage of chemical constituent present in the ashwagandha. The drug is mainly used in Ayurvedic and Unani proper preparation. So these ashwagandha roots, then leaves can be used for the preparation of the Ayurvedic medicinal medicines. Then vitaferin is a chemical constituent, contains the antibiotic and anti-tumor properties. It is used for the curing the carbuncles in the indigenous system of the medicines. Then paste prepared out of its leaves is used for the curing the inflammation of tubercular gland, and that its roots and the fruits for curing the skin disease. So it's having the antibiotic and anti-tumoric properties. So the various skin diseases, then and it act as a inflammations, means for जे जळजळ होते हाताचे किंवा जे स्किन डिसीजेस होतात आपल्याला, त्याच्या क्युरिंग साठी आपल्याला हे आपण अश्वगंधाचे जे काही रूट्स आहेत त्याची पावडर वापरली जाते. त्यापासून बनवलेले औषधांमध्ये ती पावडर वापरली जाते. सो अगेन दी bronchitis and the ulcers are also curing with this constituent. Then it is used as a aphrodisiac, then remunerative, tonic, diuretic, hypotonic, sedative, and restorative. Useful in the rheumatism, cough debility from the old age, then diprosy, then general weakness. So these are the diseases which can be cured by this ashwagandha root powder. In addition to alkaloids, roots are reported to contain the starch, then reducing the sugar, then hydrocolonatics, then glycosides, still methanol acids can be present in these as a neutral compound. Then free amino acids are identified in the roots, including the aspartic acid, glycosine, triosine, alanine, proline, tryptophan, glutamic acid, then cysteine. These are the amino acids which are presented in this chemical constituent. Then berries contain the milk coagulating enzymes, esterase, free amino acid, fatty oil, essential oils, and the alkaloids. Amino acid presents are the proline, hydroxyproline, valine, trilene, aspartic acid, glycine, asparagine, then cysteine, and glutamic acids are also present in this chemical constituents. So the origin and distribution, the ashwagandha is found wild in the grazing ground in the Mansar, which is located in the MP, Madhya Pradesh, and the forest land in the Bastar district of the Madhya Pradesh. So the mainly origin is of this plant ashwagandha is in the MP. So all over the foothills of the Punjab, then Himachal Pradesh and western Uttar Pradesh, then Himalaya range. It's found in the wild in the Mediterranean regions of the North Africa. Then crop is cultivated in area about 4,000 hectare in India, mainly in the drier parts of the Mansa, then Nimach and the Jawad Tehsil of the Mansar district of the Madhya Pradesh. So mainly found in the Madhya Pradesh, then range of the UP, some part of the Punjab, Rajasthan. So most of the dry area is found with this plant. Then Punjab, Sindh, Rajasthan, and the South India. In South India, the Karnataka, its cultivation has been reported in the Mysore district. So this is the distribution area where this plant can be found or the cultivated. Then area and production. So the ashwagandha is cultivated in 5,000 hectare in India, predominantly in Madhya Pradesh. So it is originated in the Madhya Pradesh. So MP is the native place. We can say origin of this ashwagandha is MP. So the estimated annual production in 200 to 1,500 metric tons of the dry root. So the important part of this ashwagandha plant is its roots. So medicinally important part of this ashwagandha is dry root. So dry root can be used for the preparation of the various medicines. Then varieties. So the variety name Jawahar Asand WS20 has been released from a single plant selection from the Jawaharlal Nehru Krishi Vishwavidyalay Regional Agriculture Research Station, Mansar. This variety has recorded the highest dry root yields constantly over the others. So this is the main variety we can say which is used for the cultivation purpose, which is the Jawahar Asagandha. Then high root and alkaloids yielding varieties, the Poshitha is released from CIMAP Lucknow, that is Central Institute of Medicinal and Aromatic Plant, which is located in the Lucknow. Then cultivation practices, how this ashwagandha can be cultivated. So soil and climatic requirement for ashwagandha: grows well in a sandy loam soil, in slightly alkaline soil with a good drainage condition. It grows better in 600 to 1200 meter altitude. The semi-tropical areas receiving the low rainfall are suitable for cultivation of this crop. The crop requires the dry season during the growing period. Then temperature between 20 degrees Celsius to 35 degrees Celsius is most suitable for the cultivation. Then late in the winter rains are conducive for the proper development of the plant roots. So this is the soil and climatic requirement for the cultivation of ashwagandha. Then land preparation. So as like our cultivated crop, this ashwagandha plant also requires the land preparation. So ashwagandha is usually grown in a field which are not well covered by the irrigation system. Means it is basically a dry season crop, or we can say a dry climatic requirement is required. So no uh, high quantity of irrigation system is required. So the field on which food crops cannot be grown profitably because of the raw rainfall can be used for the ashwagandha cultivation. So most of the dry area is suitable for the cultivation of this ashwagandha plant. So the soil of the field selected for the ashwagandha cultivation is well pulverized by plowing. The field should be leveled and pressed by using the heavy wooden plank. So the land can be suitably pulverized by the plowing. So or the clod crushing, harrowing, these kinds of the operation can be done. So the well leveled land can be prepared. Then afterwards the nursery raisings and the planting. The crop can be sown either by the broadcasting or in the line. So the line to line method should be preferred, increase the root production and also helps in the performing the intercultural operations then in required by the farmers. The seeds are usually sown about 2 1 3 centimeter deep during the June July in the nursery. Then light showers after the sowing ensures the good germination. The about 5 to 12 kg seeds are sufficient for the 1 hectare area. So this much quantity of the seeds are required to cultivate the 1 hectare area. The seeds are can be sown into the nursery, so the raising of the seedlings, and after that that seedlings are used for the transplanting into the main seed. So the 5 to 12 kg seeds are sufficient for cultivating the 1 hectare area. The seedlings of 25 to 35 days old can be transplanted in the field by maintaining the 30 to 30 centimeter spacing between the plant to plant and row to row. So the spacing for this crop is 30 to 30 centimeter. So row to row and plant to plant distance is same. So the as the ashwagandha is rainy season kharif crop, the time of its sowing should be decided by the date of the arrival of the monsoon in the area of the cultivation. So the as per the requirements of the monsoon, or we can say the monsoon is not same for all part of the India. So according to the monsoon arrival, the plantation or the sowing of this ashwagandha plant is changed to region. Then afterwards the other intercultural operation, we can say the thinning and the weeding can be done. So the seeds sown by the broadcasting are in the lines should be thin out by the hand at 25 to 30 days after the sowing to maintain a plant density of about 30 to 60 plants per square meter. So that much quantity of the plant population can be maintained. That means about 20,000 to 25,000 plants per hectare can be maintained. So this is the plant population, means in 1 hectare near about the 20,000 to 25,000 plants can be present in a 1 hectare. So the plant density to use many depend on the nature and fertility of the soil. So the on marginal land, the plant population should be kept high. One weeding at the early stage then sufficient to enable the ashwagandha plants to take over the growth. So the frequently weeding can be carried out as like our cultivated crop. So weed can be act as a competitive for the nutrient, then water requirements. So there must be a frequent weeding is necessary to avoid the competition between competition for the water and the important chemical or important micronutrients. Then manures and fertilizer application. So the ashwagandha crop does not require the heavy dose of the manures and fertilizers. In Madhya Pradesh, it is grown on a commercial scale. No fertilizers are applied and the crop is cultivated on only the residual fertility which is present in the soil itself. The studies at the indoor research station have showed no effect of nitrogen and phosphorus on its root yield. So this crop required a no any kind of fertilizer requirement. So we can cultivate it in a barren land which having a very less fertile soil can be used for the cultivation of the ashwagandha plant. So after the manures and fertilizers, every plant required the irrigation. So the light shower after the transplantation ensures the establishment of the seedlings. For the at the initial means the germination time, the seeds requires the water for the germination purpose. There is the no need of the irrigation if rainfall is at regular intervals. Excessive rainfall water is harmful to the crop. So this is a dry land crop, so the heavy rainfall is must affect the growth of the plant. So whatever natural rainfall is there in the dry area is sufficient for the growth of the plant. Then crop life saving the irrigation may be applied if required to ashwagandha plant. So if it requires the irrigation, so artificial irrigation can be supplied to the plant through sprinkler irrigation or drip irrigation. There is to be notified that ashwagandha is dryland crop and do not need the much water. So as we discussed that this is the dryland crop, so no such kind of the artificial irrigation system is required. So furthermore, the pest and disease management, so seed rooting, seed rotting, then seedling blight and the leaf blights are the common diseases affecting the ashwagandha plant. Their incidence can be minimized by the spraying the dithian M45 at the rate 3 gram per liter at the interval of the 7 to 10 days. So no much diseases or the pest can be affected this ashwagandha plant. So the only the seed rotting can be takes place and the seedling blights at the time of the nursery stage we can see the seedling blights or the drying of the or the black to brown patches are formed on the leaves of the plant. It can be known as a blight. So at the time of this stage we can it can be minimized or reduced by the spraying of the dithian M45, which is a fungicide, and the concentration is at the rate of the 3 gram per liter at the interval of the 7 to 10 days it can be used. So then harvesting and processing and the grading of these plants. So the plants start flowering and bearing the fruits from the December onwards. The crop is ready for the harvest in the January and March at the 150 to 180 days after the sowing. So this plant can be ready, or we plant the planting takes place in a June month and it can be ready into the month of the January to March. So the 150 to 180 days are required for the harvesting of the plant. So the maturity of the crop is judged by the drying out of the leaves and the yellow red berries in the plant starting in the field. So the typical maturity indices for the ashwagandha plant is the drying of the outer leaves and the yellow or the red, the berries can be turned from green to yellow or the red. So the typical maturity indices for the ashwagandha plant. So after the harvesting or before for the after the judging this maturity indices, the harvesting takes place. After the harvesting, the processing can be takes place in case of your ashwagandha. So the entire plant is uprooted for the roots, which are separated from the aerial parts by cutting the stem 1 to 2 centimeter above the crown. So the roots are then either cut transversely into a small pieces, that is 7 to 10 centimeter, or dried as it is in the sun. So whole plant can be uprooted. So the important plant part regarding to processing is the root part. Okay, so whole plant is uprooted, then roots can be cut into 7 to 10 centimeter from its stem part. So the dries as it is in the sun. About 350 kg fresh roots can be obtained from 1 acre of the land. On drying it comes to the 180 kg. So the berries should be hand plucked separately. They are dried and crushed to take out the seeds. So the dried roots entirely or transversely cut into smaller pieces have to be further cleaned, then trimmed and be graded. So the roots are beaten with a club, which removes the adhering soil and breaks off the thin, brittle and lateral rootlets. So the lateral branches, root crown and the stem remains. Roots are carefully trimmed with the help of the knife. So this is the typical roots of this ashwagandha plant. So afterwards, the grading can be takes place. So the entire produce, dried roots, is then carefully hand sorted into following 4 grades. So these are the 4 grades. Grade A, B, C, and the lower grade can be formed. So the grade A consists of the root piece of the 7 centimeter long. Diameter is 1 to 1.5 centimeter. Then roots should be brittle, solid, and pure white from inside. So this is the grade A. Then grade B, the root piece is 5 centimeter, diameter is 1 centimeter, then roots are brittle, solid, and white from the inside. Then grade C, the root pieces should be solid, 3 to 4 centimeter long, diameter is less than 1 centimeter. Then lower grade, small piece of the roots, then roots are somewhat hollow, yellowish from the inside. So this is the lower grade, or which can be used for the other purpose. So this is the your typical ashwagandha roots, which is used for the medicinal purpose, or which can be purchased by a pharmaceutical agency or pharmaceutical company. So this is the a typical flow chart which shows the ashwagandha harvesting and its processing. So the harvest at the time of the 180 days after the sowing. Afterwards, the maturity index is the drying of the leaves and yellowing of the yellow and red berries can be formed. So the entire plant is uprooted, then cut the aerial portion, leaving the 1 to 2 and above the crown. So the only root part is important for us regarding to our commercial use. Then roots are cut into 7 to 10 pieces, then dried into sun, bitten to remove the adhering soil portion. Then thin brittle lateral rootlets are also removed. Then dried roots are graded into 4. So the grade A is root pieces 7 centimeter long and 1 to 1 centimeter diameter, bitter solid and the pure white from the inside. So this is your grade A. And grade B is the say 5 centimeter long, then 1 centimeter diameter and solid and brittle inside, the white from the inside. And grade C is 2 to 4 centimeter long, then less than 1 centimeter diameter and solid inside. And grade D, or we can say the lower grade, hollow and yellowish from the inside. So these are the 4 grades which is followed in the ashwagandha plants. So this is all about your ashwagandha plant. So as we discussed about the ashwagandha plant, so if the area which having a less rainfall can be suitable for these cultivation, so you can planning for planting of this ashwagandha plant, as the commercial value of this plant is very high. Okay, so nowadays rainfall, some area rainfall is a very high constituent, because the frequency of rainfall is different from Maharashtra also. So anybody having this low rainfall area can be focused on this cultivation of this ashwagandha plant, because the medicinal value of this plant is very high. Okay, so give me attendance. Roll number 1, 2,
Present, ma'am.
3,
Present, ma'am.
4,
Present, ma'am.
5,
Yes, ma'am.
6, 7, 8,
Present, madam.
9, 10, 11, 12, 13, 14,
Present.
15,
Yes, madam.
16, 17, 18, 19, 20,
Yes, madam.
21, 22, 23, 24, 25, 26,
Present, ma'am.
27,
Present, ma'am.
28,
Present,
29,
ma'am.
29, 30, 31, 32,
Present, ma'am.
32, 33,
Ma'am, 31 present.
हो 32, 33, 33 is there, 34, 35,
Present, ma'am.
35 present, ma'am.
34.
okay, 36, 37, 38, 39,
Present, ma'am.
40, 41,
Present, madam.
for then previous batch 2018, 11, next 16,
Present, ma'am.
अजून
Present, ma'am.
11 आणि 16 अजून कोण आहे तुमच्या बॅचचं? 40 अजून 30 हा ओके 38 आणखी बस एवढेच ना आणि नंतर 2017 चे
गावित मॅडम 2017-13.
13 ओके आणखीन कोणी आहे का 2017 च्या बॅचचं? नाही ना गावित तुझ्यासोबत आणखीन कोणी आहे का रजिस्ट्रेशन ह्या कोर्सला?
आहे ना गावले आणि पाडवी आहे या
गावले
सेमिस्टरला.
आणि पाडवी का बरं ठीक
हम्म.
2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20 21 तुम्हाला हा टॉपिक किंवा हा क्रॉप खूप इम्पॉर्टंट आहे मिड सेमिस्टरला विचारलं जाऊ शकतो आणि त्याचे जे काही ऑब्जेक्टिव्ह आहेत म्हणजे केमिकल कन्स्टिट्युएंट आणि कल्टिवेशन प्रॅक्टिसेस इज इम्पॉर्टंट रिगार्डिंग टू युअर एक्झाम. सो इन दी इव्हनिंग 4 to 5 we have another lecture, so we'll discuss about the periwinkle or we can say the sadafuli. Okay, so I end this meeting.